My name's Bryce. I did my bacterial disease on Streptococcus pneumonia. The reason why I picked Streptococcus pneumonia is because of the wide history and the background about Streptococcus. I really like the medical side of diseases and how that affects your body and how it works. So the background of Streptococcus is really interesting. It first started when a strain of the Streptococcus pneumonia started affecting soldiers in World War II, and it started to kill a lot of injured soldiers, and then the ones who they didn't kill, they would bring it back to their families, and it started to become a really big problem. So a scientist named Frederick Griffith, he did a lot of research on it, and he took two strains. He took one with a capsule on it, which a capsule is basically like a protective layer of the bacterial strain, and one without. So he took both of these. When he injected the strain without a capsule, he noticed that all of the mice survived because our bodies are able to dispose of the strain without the capsule. But when he injected the strains with a capsule, all of the mice died. So it's really interesting about why something as simple as a capsule can cause death in all mice versus without a strain where all of them survive.
You don't need everything.
He also took a strain of the capsule without a capsule and a dead strain with a capsule, and noticed that the dead strain transferred to the live strain, and it killed all the mice, which at the time was a huge discovery for science. No one ever thought that a dead bacteria cell could transfer into a living bacteria cell. And so they were able to figure out a lot of things from that, and it was really important for science. And that's kind of where I based my project on, the capsule protein. I thought that they were really interesting. So we ran through, this is a pipeline of steps that we run through. So first we got all of this data, and we get the raw data, and then we do a thing called Chemomatic, which is a picture of how Chemomatic works. And it's basically like, this is the level of certainty that we know that the strain is. So Chemomatic, it cuts out poor strains. So when we have a strain that is way up here that you really don't know what it is, it's important to cut it out. So when we have our data, we know that it's good and it's quality and it is what we think it is. So we ran through Chemomatic, then we assembled it back together because the way Chemomatic, it cuts them. So you need something to put them all back together, all the good strands. We ran a lot of quality and annotation, which are things like this, where you compare a strain. In this case, it was strain 3. So we compared strain 3 to a reference strain. And what that does is basically, if it's close like this one is, you know that you have the correct strain that you're working on, which is important because you don't want different strains that you aren't sure or have a different things in it. So this is also from annotation, which it just compares 7 strains together, how they're different, how they're similar, their size, the number of things that they have in them, and this is just another thing that we did to make sure that all of the strains that we had were closely related to each other. So we did a lot of stuff to make sure that we had the correct bacteria and everything, all the data was good. So, and then once we get to Rory gives us an output folder, which from there we can use to see this, which compares all the strains together. And all this blue is the core genome, so that's genes that every Streptococcus pneumonia strain shares together. All of these 20 share, which is important to see because when we see that, we can open up a output folder that we get, and it shows which strains have which. So from here, this picture is a capsule synthesis protein, and from here you can see that all 20 of the 20 strains have that. So it's something that would be in the core genome. When you figure that out, since I've been looking at capsule synthesis protein, there was 3 proteins that we looked at that were in the 20. So there was the capsule synthesis ACB, which 20 out of 20 of them had. There was a cap A, which 17 out of 20 had. There was another cap A, which 4 out of 20 had, and a cap B, which 6 out of 20 had. And from there, a little bit of research was done, and I found out that it has to do with the virulence. So if a strain has a cap A protein, then when we look at this, the phagocytosis, when it goes to destroy them, it's a little easier to destroy one with only a cap A or a cap B, but when the strain has both cap A and B, that's when it is a really strong bacterial strain and it causes death in a lot of people. So, and that's all the research I did.